Welcome to Lecture 7.3, in which we will go over the light-independent reactions, also known as the Calvin cycle. This is a summary of the light-independent reactions, and similar to the citric acid cycle, it involves combining two molecules together at the beginning, and one of the molecules is regenerated at the end to be used in another round. I will point out the parts that you should know for this class as we go through the video. Our learning objectives for this video are to describe where the light-independent reactions take place and the overall inputs and outputs, describe the three main steps of the light-independent reactions, understand the role of Rubisco, RUBP, and G3P, and learn more about the relationship between the light-dependent and light-independent reactions. The light-independent reactions take place in the stroma of the chloroplast and can be broken down into three stages: fixation, reduction, and regeneration. It starts with taking carbon dioxide and combining it with a 5-carbon molecule. During this process, the ATP and NADPH produced by the light-dependent reactions is used. This means that even though these reactions don't directly depend on the light is still necessary to produce the ATP and NADPH that's needed for the light-independent reactions to occur. After several cycles, glucose is produced, and the 5-carbon molecule regenerated. Since the carbon dioxide is being used to make the glucose, 6 cycles are needed to obtain the 6 carbons needed to make one glucose molecule. This diagram shows the amount needed of each molecule to make half a molecule of glucose. So you would just multiply everything by 2 to get the numbers you need to make a full molecule of glucose. However, for this class, you just need to know the number of cycles it takes to make glucose. Fixation, the first step of the light-independent reactions, involves taking a 5-carbon molecule called ribulose 1,5-bisphosphate, abbreviated as RUBP, and combining it with carbon dioxide. This produces a 6-carbon molecule that is immediately split into two 3-carbon molecules, 3-phosphoglycerate. The enzyme that performs this reaction is called ribulose 1,5-bisphosphate carboxylase/oxygenase, abbreviated as Rubisco. We saw some decarboxylases in cellular respiration, which remove carbon dioxide. Carboxylase does the opposite by adding carbon dioxide. This process is referred to as carbon fixation because Rubisco is fixing carbon dioxide in place, preventing it from escaping. As an analogy, you can think of RUBP as a nail that will keep the carbon dioxide in place, and Rubisco is the hammer that drives the nail in. For both RUBP and Rubisco, I am not expecting you to memorize what the full names are. I am expecting you to know that RUBP is the 5-carbon molecule and Rubisco is the enzyme that is used for carbon fixation. After the 3-carbon molecule is produced, ATP made from the light-dependent reactions is used to phosphorylate it. You don't have to remember the names of these molecules, but you might recognize this as a reverse of a reaction that occurs in cellular respiration. Next, NADPH made from the light-dependent reactions is used to reduce the 3-carbon molecule. This produces G3P. Again, this reaction is essentially the reverse of a reaction that occurs in cellular respiration. Once the G3P molecule is produced, it can do two things. One is to produce a glucose molecule. 2 molecules of G3P are needed to produce one glucose molecule. This is done in a series of reactions called gluconeogenesis, which essentially means creating new glucose. Similar to how a few of these steps are the reverse of one seen in glycolysis, gluconeogenesis creates glucose using the same beginning steps of glycolysis but in reverse, with a few exceptions. The glucose that is made can be used to make disaccharides and polysaccharides. Other G3P molecules are used to regenerate RUBP in a series of reactions that we won't go over, and the RUBP can now be used to perform another cycle. Ultimately, photosynthesis and cellular respiration are closely related processes. They have many similarities, such as the molecules involved and the electron transport chain. Remember that plants and other autotrophs act as producers that take in carbon dioxide and water to produce carbohydrates and oxygen. Heterotrophs act as consumers that eat the producers or other consumers, then break the carbohydrates back down into carbon dioxide and water, and the circle of life continues. In this video, we learned that the light-independent reactions take place in the stroma. At the beginning of the reactions, the enzyme Rubisco is used to fix carbon dioxide to RUBP, which produces two 3-carbon molecules. ATP from the light-dependent reactions is used to phosphorylate the 3-carbon molecule, and then NADPH from the light-dependent reactions is used to reduce it to G3P. The G3P can be used to produce glucose, or be used to regenerate the RUBP. The plants themselves can break down the glucose they create through cellular respiration, or consumers such as us can eat these plants to obtain and break down the nutrients for energy. That's it for photosynthesis. Thanks for watching.